Routine clinical brain scans from millions of patients can train AI models that reach specialist-level diagnostic accuracy. A foundation model trained on 5,024,000 clinical CT and MRI series reached state-of-the-art performance on diagnosis tasks. The training used only routine hospital scans rather than curated research datasets. The model also supported preliminary report generation inside real health systems. It outperformed models built on public internet and medical data. The study appears in Nature Medicine. The work shows that everyday hospital imaging contains enough signal to train specialist-grade diagnostic tools without extra curation. A second model, called PRISM-2, was trained on 2.3 million whole-slide images paired with 14 million clinical question-answer exchanges. PRISM-2 achieved clinical-grade cancer detection without task-specific fine-tuning. Clinical dialogue supervision provided the key signal that allowed generalization across pathology tasks. Performance held across multiple cancer types and staining methods. Further validation will assess integration into diagnostic workflows. Now shifting to a very different area of research. Researchers built a detailed spatiotemporal omics atlas that tracks how primary tumors evolve into metastatic disease across space and time. The atlas maps cellular states, gene expression changes, and microenvironment shifts at successive stages of progression. This approach reveals previously hidden transition points where cells acquire metastatic traits. The study appears in Science and provides a framework for identifying intervention windows before full metastasis occurs. Follow-up work will test whether the mapped pathways can be targeted in model systems. On a different note, large stores of organic carbon in Arctic permafrost can enter the ocean through thaw or coastal erosion. New measurements show the ocean keeps the majority locked away in marine sediments instead of returning it to the atmosphere. The study quantifies how much carbon stays in sediments versus what escapes. This changes estimates of the permafrost carbon feedback to climate. Next measurements will track seasonal and regional variation in retention rates. The work appears in recent reports summarized by phys.org. Now here is something most people get wrong about thawing permafrost. Most people assume that thawing permafrost automatically sends large amounts of carbon into the atmosphere as greenhouse gases. In reality, when permafrost carbon reaches the Arctic Ocean, much of it stays locked in marine sediments rather than returning to the air. Right now, as permafrost continues to thaw along Arctic coasts, microorganisms in the water column and seafloor determine how much carbon escapes versus how much gets buried. One memorable figure from recent measurements is that the ocean retains the majority of this carbon instead of releasing it. This changes how climate models should account for the permafrost feedback. A practical takeaway is to watch for improved regional estimates that separate what stays in the ocean from what reaches the atmosphere. Those numbers will refine projections of future warming. From environmental carbon dynamics, we move to a different kind of preservation. Scientific papers present a linear path from question to result, omitting failed experiments, redirected projects, and informal conversations that shaped the work. A new seminar approach records the tacit knowledge of retiring field founders before it disappears. The method reconstructs the actual sequence of decisions and dead ends that never appear in print. It aims to preserve context that influences how future researchers interpret the literature. Organizers plan to test the format across additional disciplines. A UC San Diego team added trace europium to hydroxyapatite, the main mineral in teeth and bones, to mimic calcium behavior. The modification allowed better tracking of the mineral's structure and stability under different conditions. The approach could support medical imaging applications that require clear visualization of bone-like materials. The researchers demonstrated the method in controlled laboratory settings. Further tests will examine performance inside living tissue models. University of Plymouth researchers compiled evidence of major changes in marine plankton and pelagic habitats around the UK. The MCCIP report documents altered community composition linked to warming and other climate factors. These shifts affect the base of the marine food web and carbon cycling. The data come from long-term monitoring programs that allow detection of trends over decades. Continued surveillance will determine whether the changes accelerate or stabilize. From that effort at preserving research context, we turn to another effort that uses genomic context to settle a historical question. Viral genomes recovered from Chilean mummies match European smallpox strains and show no evidence of pre-contact circulation in the Americas. The data provide the first direct genomic support for introduction by European contact. The sequences also reveal how the virus adapted after arrival. This finding settles a long-standing debate about the disease's New World history. Additional ancient samples will test whether other pathogens followed similar patterns. An international team, led by the University of Queensland and University of Michigan, uncovered a process that increases mutation rates at specific genomic sites. The work links the mechanism to elevated disease risk in carriers of particular variants. Experiments in cellular models confirm the pathway's role in generating the mutations. The finding adds a new layer to understanding why some genetic conditions cluster in families. Follow-up studies will examine whether the process can be modulated. From historical pathogen genomics, we shift to a contemporary diagnostic challenge in neurology. Queensland Brain Institute researchers used high-speed eye tracking to re-examine oculomotor deficits in progressive supranuclear palsy. The data contradict a long-standing clinical belief about the timing and pattern of eye movement abnormalities. The results suggest earlier or different diagnostic markers than current criteria assume. The study included patients at multiple disease stages. Larger cohorts will determine whether the revised markers improve diagnostic accuracy. A systematic review of published cases found Wernicke's encephalopathy occurring after semaglutide use for obesity. The review followed PRISMA guidelines and compiled clinical details from available reports. Thiamine deficiency appears central to the observed neurological symptoms. The authors note that rapid weight loss may contribute to the deficiency. Clinicians are advised to monitor at-risk patients for early signs. The work appears in recent reports summarized by Wiley Online Library. One trial found metformin halved the risk of long COVID. A second trial reported a 40% reduction with Paxlovid. Both studies used randomized designs in patients with acute infection. The results add to evidence that early antiviral or metabolic intervention can alter post-acute outcomes. Effect sizes differed between the two agents. Larger confirmatory trials are underway. FCD11 blocks the interaction between SET1A COPC and accessory proteins, lowering H3K4Me3 methylation. In cell lines and mouse models of breast cancer, the compound decreased tumor size and extended survival. ChIP-seq confirmed selective inhibition of the target complex. The work establishes a first-in-class lead for cancers dependent on SET1A activity. Preclinical safety studies are the reported next step. Before we go, keep an eye on further validation of the eye tracking markers for progressive supranuclear palsy. That's Planetterrian Daily for today. If you enjoyed this, a rating or review on Apple Podcasts or Spotify really helps new listeners find the show. I'm Patrick in Vancouver. Thanks for listening, and I'll see you tomorrow. And if you'd rather watch than listen, find us on YouTube at NARA Network. Links in the show notes. This show comes to you from the NARA Network. If you want another sharp daily briefing, Models and Agents has you covered. Your daily briefing on AI models and agents. All our shows are free at naranetwork.com. New to the network? naranetwork.com Start Here picks the right daily briefing for you in about 2 minutes. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.